Welcome, ladies and gentlemen, to the first lesson of Unit 2, talking about cells and cell functions. Also, welcome to our first meme, including my cats. That's the fat one, by the way. So we're going to be talking about cells, more specifically the parts of cells. But first, let's do a little general information about cells. All cells do the following functions. They maintain homeostasis, they make proteins, and then they have to be able to pass on traits to the cells that they then give rise to. Note, when we're talking about the products or the offspring of a cell, we call them daughter cells. Yes, it's slightly sexist. I don't know why. So the basic units of structure and function in a cell are organelles. The way that I like to think of it is that the ending L actually is a suffix that you can put onto the end of a word to mean little. So what you could do is you could think of this as what I used to tell my freshmen when I taught freshman bio, is that it's a suffix like adding ito to the end of a word in Spanish. So it's like a little organito. It's a little organ that does kind of a job for a cell that an organ would do for your body. Now, keep in mind, this is an analogy. Some of these don't have full counterparts in the human body because they're going to do different tasks needed to keep a cell alive. All cells have the following structures. They have DNA. That's their genetic material. They're going to be surrounded by a cell membrane. They're going to have ribosomes to make proteins, and they're going to have a cytoskeleton that helps them keep their shape, also helps with movement, and filling their cells is going to be cytoplasm, which includes a liquid called cytosol. So any cell on this planet has to have these 5 elements. It must have all of them. So if you're looking at a cell, if you're being asked to identify a cell, and you get a mention something along the lines of it has a cell membrane, it has ribosomes, it has a cytoskeleton, that's not going to tell you what specific type of cell you're looking at. All it's going to tell you is, yes, you're looking at a cell. There are other organelles that we'll look at that will kind of give you more of a clue to the specific type of cell that you're looking at. So let's look at these types of cells. Ultimately, you can take all cells and divide them into 2 separate groups. They can be prokaryotic or eukaryotic. This should be a fairly familiar concept from freshman biology. You should hopefully remember that prokaryotic cells have no nucleus, and that this word is synonymous, prokaryote, with a bacterial cell. So all prokaryotes are bacterial, all bacteria are prokaryotes. We can take all of the rest of life on the planet and divide it, for the most part, into 3 major categories: animals, plants, and fungi. There are also protists, which used to form their own separate grouping, but now, based on genetics and kind of their evolutionary relationships, they're being classified more as akin to animal, plant, or fungal groupings, though they're still not true animals, plants, or fungi. And we may talk about that more at a different time. If not, if this truly interests you, feel free to come talk to me during study time. All eukaryotic cells have a nucleus, and this does include protists too. Not only do they have a nucleus, they have a number of other organelles inside, which we call membrane-bound organelles. So they are very compartmentalized and they are organized. So, like I said, prokaryotes don't have a nucleus. They don't have a lot of compartments within them. Their DNA tends to be basically continuous with the rest of the cell, with the cytoplasm. It tends to be located in a region in the center of the cell called the nucleoid, but it's not physically separated by a membrane. And there are 2 major groupings that contain prokaryotic cells. These are domains, bacteria, and domain archaea. Eukaryotes, on the other hand, do have membrane-bound organelles. They tend to have certain structures in common. For example, all eukaryotic cells have a nucleus. All eukaryotic cells have endoplasmic reticulum. We have Golgi, we have mitochondria, and then depending on the subtype of eukaryotic cell you are, you might have additional specialized organelles as well. All of us who are eukaryotes fit into domain Eukarya, which contains, at least at this point, 4 kingdoms: Protista, Fungi, Animalia, and Plantae. Like I said, though, Kingdom Protista will probably not be around for very much longer, if it's even still technically recognized by most biologists. Prokaryotic cells all have a cell wall around them. A lot of them will contain these molecules called peptidoglycans, and if you look at the word, you can kind of figure out what type of molecule it is. They're actually kind of a hybrid biomolecule. That peptide in the front, peptido, means that they have a protein portion. So they're going to have a portion that's amino acid. The glyco part in glycan means sugar. So these are actually what I kind of like to call Frankenstein molecules. They've got bits and pieces of different categories of biomolecules. So they're going to be made of amino acids joined to sugars chemically. Some bacteria will also have a specific separate outer membrane. And then you can also have a capsule surrounding bacteria. This is kind of a slimy layer. It's made of polysaccharides. And if you'll remember, polysaccharides are carbohydrates. And it's a protective layer that can surround certain bacteria, and when bacteria are surrounded by a capsule, they can survive very harsh conditions without dying, including dehydration, lack of nutrients, and infectious bacteria that form capsules are particularly problematic because when they're in a capsule, they can't be killed by a lot of traditional methods. So sometimes heating will not kill them, and antibiotics will not be able to kill them either. There are differences in structure between the cell walls of different bacteria. If you end up studying bacteriology or immunology, you may learn much more detail about the molecular composition of these. There are also differences between eubacteria and archaebacteria, and we'll probably talk about those just a wee bit more when we are in evolution. So these are a couple of images of bacteria. You can see the prokaryotes don't have a ton of structures inside of them. You will generally see the nucleoid. It looks like a kind of darker spot in the center of the cell. You'll see the membrane, there isn't one, my pardon. The cellular membrane, and there are often external structures as well, things like cilia or flagella that help bacteria move. There are bacteria that are capable of performing photosynthesis, and if you stop and think about it, you may wonder, how can they perform photosynthesis if they don't have chloroplasts? In actuality, the ancestor of the chloroplasts that are found in plants was probably a photosynthetic bacterial cell. What they do have, instead of having a separate membrane-bound organelle, is they have infoldings of their of their cell membrane, and these infoldings will contain the molecules needed for photosynthesis, and we'll talk a little bit more about this when we talk about photosynthesis in Unit 3. Eukaryotic cells are defined by the fact that we have membrane-bound organelles, and because of these membrane-bound organelles, it overcomes quite a few limitations on, for example, cell size and also compartmentalization. The reason why we are able to be multicellular is because of the specialization that this kind of increased complexity of eukaryotic cells allows for. So, for example, in the cytoplasm, the pH is usually fairly close to neutral, around 7 to 7.4, but you have these digestive compartments called lysosomes where the pH is much more acidic because that helps in the breakdown of molecules. On the other hand, if you had this acidic pH exposed to the rest of the cell, it would start breaking down the rest of the molecules that the cell needs to function, including necessary proteins, and might even start breaking down organelles that are needed to keep the cell alive. Instead, you can compartmentalize them into this lysosome where they're available to do the work of digestion, but they're not going to damage the rest of the cell. So you'll see many images of various organelles. Here's an image of a lot of the organelles that are found in an animal cell. I highly suggest you pause this slide and the next slide, take a look at the organelles, especially because you have both electron microscope images and a diagram here. You will see diagrams on the AP test. Some of them may be electron microscope images, like the ones you see on this slide. Some of them may just be drawings, but you need to get used to being able to identify the organelles from both of these. Here are organelles also found in a plant cell. Some of these are found only in plant cells, for example, the chloroplast. The cell wall is not found in animal cells. Others are found in both animal and plant cells. For example, the Golgi apparatus, the free ribosomes, and the smooth endoplasmic reticulum, and we'll talk about each of these in more detail. First, let's talk about the organelles that all cells contain. So all cells contain cytoplasm. Cytoplasm is basically the space outside of the nucleus and inside the cell membrane. It contains all the organelles that are not the nucleus and the space between, which is filled with not only a large number of molecules, but also a fluid called the cytosol. The cytosol is where a lot of chemical reactions happen. In fact, in Unit 3, when we talk about cell respiration, we'll talk about the fact that a very important first step of cell respiration happens in the cytoplasm. What you're seeing here is not just like some sort of abstract painting. What you're seeing is an image that's supposed to be fairly representative of what the molecules in the cytoplasm look like. As you can see, it's not a lot of empty space like what you see in most diagrams and pictures and images of cells. Instead, the cytoplasm is very, very full. It's probably more viscous than water, meaning it's more jelly-like rather than free-flowing like water, because it's got a lot of molecules dissolved in it. So what you're seeing here are kind of 3D images of various proteins. This big protein right here that's stretching like a long chain, that's going to be a microtubule, which we'll talk about in a little bit. You'll see some smaller chains, which will be actin filaments or intermediate filaments. You'll see these kind of globular proteins that will have various tasks inside of the cell, and they're all just kind of floating around. But I do want to point out that it's a very crowded environment. So if you're relying on diffusion to move stuff around in a cell, anything that's trying to diffuse will have to get through all of these molecules that are crowding the space. Let's talk cytoskeleton. So the cytoskeleton does the same job for a cell that our skeleton does for us. It helps anchor. So, for example, various organelles may be anchored to the cytoskeleton to keep them in place. It gives structure to a cell. So when cells are forming these more complicated cellular structures, that may be held in place by these cytoskeletal elements. And it can also change shape and allow for motion. So, for example, there are motor proteins that will associate with the cytoskeleton and help either move things around within the cell or move the cell itself. This is seen very commonly in things like lymphocytes or white blood cells that are able to move and change their shape. You won't see this so much in a lot of cell types in the human body or in animal bodies that don't change shape. You won't see this in plant cells really, but even the structures that give mobile cells the ability to move around also contain elements of the cytoskeleton, for example, flagella and cilia, which allow cells to swim through fluids. The cytoskeleton elements also help interact with the extracellular structures that help keep cells in place. This is especially important if you, like us, are multicellular and you want to keep your cells arranged in a particular arrangement. There are multiple components that make up the cytoskeleton. They have some things in common. They have some things differently. They're all made of subunits. So we'll mostly talk about the subunits of microtubules and actin filaments because intermediate filaments can have a couple of different subunits. But microtubules and microfilaments exhibit this quality called dynamic instability. If you look at it, those two words that make up the name are kind of opposites of each other. If something is dynamic, it's constantly changing. sorry, they're not opposite of each other, they're the same. Dynamic means it's constantly changing. Unstable means it's constantly changing. So what happens is these things can be built and broken apart very quickly. It's just a matter of polymerizing or adding more monomers onto a microtubule or onto an actin filament to make it longer or to make it shorter. So because of this, the cytoskeleton is a bit more flexible than our cell skeleton. Your bones don't tend to change shape dramatically. However, the microtubules that are making up part of the cytoskeleton can be broken down and reshaped as needed. So you can very quickly assemble or break down parts of the cytoskeleton. Another thing, the elements of the cytoskeleton, these three different types of fibers, are directional. They have a positive end and a negative end. Things can only be added, monomers can only be added to the positive end, and they have to be removed from the negative end. So there's some directionality there. So let's talk about each of the different types of fibers. Microfilaments are formed of these repeating units of this protein called actin. They're very good at bearing tension, pulling forces from ripping the cell apart. They're very useful for not only supporting the cell shape, but also for moving parts of cells. They participate in something called cytoplasmic streaming. If you've got imaginary spare time, I would look up videos on cytoplasmic streaming just because it looks kind of cool. It's basically stuff floating around, almost like a current inside of your cell. They're also important in cell division as well. And actin filaments are important in the contraction of muscles too. They also make up the cores of these structures called microvilli, which are important in your intestines. They increase your surface area to make sure you can absorb a lot of nutrients in your intestine in a very short period of time. Intermediate filaments tend to be made up of various members of a family of proteins called keratin. Notice, once again, we're looking at proteins. All elements of the cytoskeleton are made of proteins. These tend to be more permanent than the other two types of fibers. They're not built and broken down very quickly, so they tend to be more long-term permanent structures. They help maintain the shape of the cell, and they will anchor cell structures. Microtubules are a hollow tube made of these tubulin monomers. So we have two types of proteins. They're both types of tubulin, obviously named for the fact that they're in microtubules. This forms a rigid internal skeleton. It's also the microtubule network inside of a cell is what pulls apart chromosomes during cell division. It forms the core of cilia and flagella, so that way they can help move a cell around. And they're involved with these really cool proteins called motor proteins that will move sometimes entire organelles around inside of a cell. In fact, there's a very special group of microtubules called centrosomes or centrioles. Some in plant cells, for example, they're called something different. They are called microtubule organizing centers, or the MTOC, and they are very important because they are what form the protein spindle, those fibers that will catch chromosomes and separate them during mitosis. So very essential to a very fundamental process. They also form the internal structure of cilia and flagella as well. So you have microtubules. They're arranged kind of around a core of microtubules with some motor proteins involved that help move cilia, make them kind of wave back and forth. Same thing with flagella, allow them to kind of form this wave-like motion that helps propel cells. Like I said, there are these cool motor proteins that are associated with them. I don't anticipate you needing to know anything about them, but one of them happens to be my favorite molecule in the whole body, so I thought I'd introduce you. So dynein and nexin are involved in basically the movement of cilia. However, my favorite molecule in the body is kinesin. Kinesin is this molecule right here that kind of looks like a pair of feet, and that kind of matches its function because it actually literally walks things down a microtubule. We're going to watch some videos in class where we're going to look at some kinesin molecules moving, and you're going to see they actually look like little feet walking along, which is really cool when you're a nerd. Ribosomes are made of RNA, a specific type of RNA called ribosomal RNA, real creative I know, abbreviated rRNA, and they're bound to small proteins. So when we were talking about RNA world and we were talking about the fact that RNA can catalyze some chemical reactions, just like enzymes do, one of the kind of examples of that we look at is the fact that the RNA that's in ribosomes helps to perform a chemical reaction that builds proteins. There is a large unit and a small subunit. They are not covalently bound together. They associate with each other around an RNA. When they're not bound around an RNA, they will separate. Their function is to take mRNA and use it to translate that sequence of nucleotides, the A's, the C's, the T's, and the G's, or in the case of RNA, A's, the U's, the C's, and the G's, into a sequence of amino acids. So they're responsible for going from a nucleotide sequence and from that, they build the primary structure of proteins. Remember, the primary structure of proteins is basically the chain of amino acids and the identity of those amino acids in their specific order. So that is built by ribosomes. Ribosomes are in prokaryotes just found floating in the cytosol. In eukaryotes, they can either float in the cytosol or they can be attached to another structure called rough endoplasmic reticulum. They can also form these neat little units called polyribosomes, where basically you have one mRNA and you've got like 10, 20, however many ribosomes, all translating it at the same time. This means that you can use one mRNA and you can use it to make 20, 30 copies of a protein all at the same time, which makes it much faster. You don't have to wait for an mRNA to feed through one ribosome before you can use it to make another protein. So here's just showing you some ribosomes that are attached to rough ER in this electron microscope image. For example, these guys right here are ribosomes, and we'll try and look at some videos and some images in the future. Now, let's look at the organelles that are only found in eukaryotes. So these are not found in bacterial cells or prokaryotes. Remember, prokaryotes and bacteria are the same things. So, a eukaryotic cell has both cytoplasm and what we call nucleoplasm. Plasm literally just kind of means like stuff or goo. So cytoplasm is literally the cell stuff, and nucleoplasm is literally the nucleus stuff. They are separated from each other by the nuclear membrane, and the cytoplasm is everything outside the nucleus and inside the cell membrane. So remember, it includes all of the organelles that are not in a nucleus. Remember, since prokaryotes don't have a nucleus, it's pretty much all cytoplasm. So first we're going to look at what we call the endomembrane system. It's pretty much a continuous chunk or continuous chunks of membranes that can interconnect with each other. And let's move to the diagrams because that might make it more understandable. So a lot of the protein synthesis and protein packing that happens in a cell happens in these structures, and it includes things like the nuclear envelope, which is the outside boundary of the nucleus, endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles, and the plasma membrane. So these guys can all be interconnected by these small basically balls of membrane called vesicles. I think of them like little purses or little sacks for the cell, in that they can be used for any purpose. You can basically pinch a vesicle or a little ball of membrane off of another organelle, and it becomes its own little free-floating structure that can move around the cell and kind of ferry stuff in between these different organelles. So let's just take a quick look at how proteins are synthesized and packaged in the cell. Everything starts in the nucleus, where you have the instructions for making proteins or the instructions for the primary structure of proteins stored in DNA. This is where you make an RNA copy, which then can, in eukaryotic cells, travel outside of the nucleus into the cytoplasm, where it attaches to a ribosome, which starts translating a protein. Now, a lot of ribosomes in eukaryotic cells are attached to the endoplasmic reticulum, where these proteins will kind of be dumped into the endoplasmic reticulum. They can be concentrated, they can be modified, they can be chaperoned into folding into the right configuration. Some of these proteins will actually become part of the cell membrane. They'll be incorporated into the membrane of the endoplasmic reticulum, and then these proteins can be shipped out by vesicle to the Golgi. I'll explain the apparatus body complex in a moment, which can then modify these proteins and then prepare them to be released from the cell if that is their final destination. Obviously, there are going to be proteins that stay in the cell. We're just talking about if you have proteins that are being secreted or released from the cell, where they're going to go. So let's look at images of this. So here we have the nucleus, we have the rough ER where you have some ribosomes attached. This is a vesicle starting to bud off. It just kind of pinches off from the membrane making up the ER, and then it can travel to another location, for example, the Golgi. When it does, that vesicle can basically melt into the membrane that makes up the Golgi. Depending on what modifications need to be made to it, it can be sent to any of the little pancake-looking sacks of membrane that you have making up the Golgi. It can move from those to another, or it can be released from what's called the trans space of the Golgi, and then it can meld or kind of fuse with the plasma membrane, where then you can release the contents outside of the cell. So let's talk nucleus. The nucleus is all about DNA. It stores it, it protects it. It's also where anything having to do with DNA has to happen, including transcription or making copies of RNA. And it also contains the nucleolus or nucleolus, you can pronounce it either way, which is where our RNA or ribosomal RNA is transcribed and where our ribosomes are made. So where are ribosomes made? They're made in the nucleus. The nucleolus or nucleolus, and I will go back and forth between saying it either way, is a darker spot inside the dark spot that is the nucleus. The nucleus tends to be darkly colored in most images because when you look at a cell under a microscope, you add colors to it, which are stains or dyes, and once you add those colors, they tend to mostly concentrate in certain structures, including the nucleus. So when you're looking at a microscope image of a cell, the nucleus tends to be very darkly colored. So the membrane of a nucleus is called the nuclear envelope, and it's got a number of holes in it called nuclear pores. These nuclear pores will allow the transport of molecules between the nucleus and the cytoplasm, but they can also act as regulators of what can go in and out of the nucleus, which is a good thing. You don't want just any molecule having access to your DNA. So the nucleus contains chromatin, which is DNA that's been kind of condensed around proteins. It's not fully coiled up like a chromosome that you see in a dividing cell, but it is loosely organized. And the nuclear membrane, importantly, is continuous with the endoplasmic reticulum. So those two structures are physically connected to each other. And to look at why, we will also look at kind of the evolution of the endomembrane system when we talk about evolution more. We'll also look at an animation in class that will help clarify a little bit too. So here are just some electron microscope images showing various parts of a nucleus. You can see nuclear pores here. So each of these little flower-looking structures is a nuclear pore. Here's kind of just showing you in diagram form, and you can see that the membrane of the ER is continuous, meaning physically connected to the membrane of the nucleus. All right, speaking of ER, let's talk about the endoplasmic reticulum. The endoplasmic reticulum makes up a large amount of the membrane in any eukaryotic cell. We can talk about the outer membrane of the endoplasmic reticulum or the space inside. When we are talking about spaces inside hollow areas, you'll see this general term lumen. You can have the lumen of many things. You'll have the lumen of the Golgi. You'll also can have the lumen of organs. For example, your intestines have a lumen as well. It's just an interior space. This is also called the cisternal space at times. So if you see either of those, you're talking about you can be talking about the ER. There are two different subtypes, rough ER and smooth ER. They have different appearances and different functions. It is important that you know the difference between them. So let's talk about their difference in function. Rough ER, big thing is it's involved in protein synthesis. So if you remember nothing else about the rough ER, remember protein synthesis. Remember, it's got ribosomes attached to it. That's what gives it its name, because the ribosomes that are attached almost look like beads stuck on the surface, and that gives it this kind of rough, bumpy appearance. You can not only have proteins made here, they'll be modified. They can be tagged to be delivered to certain areas of the cell. They can also be modified. So a lot of proteins will have things added to them that are not amino acids. For example, if they get carbohydrate groups added to them, they will become glycoproteins. Remember, glyco means sugar. Anything that's going to be released from the cell, and most things that are going to be incorporated into the cell membrane are going to pass through the rough ER. It's also an area where you produce some membrane. The smooth ER has a number of different functions. You're not going to need to know which function goes with which cell, but I did want to go over a couple of the general functions it has. So smooth ER can detoxify, meaning it can break down poisonous molecules. It can help break down glycogen in animal cells. Glycogen is a polysaccharide, which means it is, hopefully you can answer the fact that it is a carbohydrate. You can also make lipids, including phospholipids here, steroids as well. You can store things in smooth ER. For example, in certain types of cells, you'll store calcium, and you can also modify carbohydrates and steroids here as well. As far as the structure, a rough ER looks like these kind of flattened folds of membrane, and like I said, it's got a kind of rough appearance. Smooth ER, on the other hand, looks like a bunch of interconnected tubes. So here you see both rough and smooth ER in both diagram and electron microscope image. So notice smooth ER not only doesn't have ribosomes attached to it, please excuse the cat sounds, it kind of looks more like spaghetti. It looks like a bunch of goopy strands, whereas the rough endoplasmic reticulum has a bunch of ribosomes attached to it. It kind of looks like somebody flattened it, because it looks like these kind of stacked, flattened sacs. This is the appearance of the smooth ER in a electron microscope image. So you can see kind of, you're looking at those tubes like they've been cut in half, and on the rough ER microscope image you can see the little ribosomes that are attached to it. Let's talk about the Golgi now. So depending on the textbook you look at, or what specific information you're looking at, I've seen it referred to as Golgi apparatus, Golgi body, Golgi complex. They are all the same thing. So I mostly will just refer to these as the Golgi. Occasionally I'll call them the Golgi ABCs. I've tried to find out if there's a general consensus among biologists which one of these terms you should use, and honestly I haven't found one yet. So just keep in mind, most of the times when you see the term Golgi, you're talking about this organelle. What its job is it modifies proteins. So it takes proteins that are made in the ER, they're transported there by vesicles, and they're going to be modified here. You can also have certain other macromolecules made here, and it's going to sort them. It might concentrate them, meaning like kind of build them up to higher concentration so that you have a lot more of them in a certain space. Packages these guys into transport vesicles so they can be sent around the cell. It can modify proteins so that they have carbohydrates added to them, so they're glycoproteins. And if you are in a plant cell, they make the polysaccharides that are needed to make plant cell walls. And remember, what is a polysaccharide? It is a complex carbohydrate. So the structure, it's made of these flattened sacs called cisternae, very similar to what you saw in rough ER, and there are small vesicles. I like to think of it like a stack of really melty looking pancakes, because if you looked at it in 3D form, that's what it would look kind of like. It does have some polarity to it, meaning not like charge, but meaning that it's got specific sides. So there is a side that specifically faces the ER. It's called the cis face. Cis means same. So this is facing other organelles that are part of the same cell. Trans means across. The trans face is going to be the side that faces the plasma membrane, and it's the side that's going to mostly produce vesicles that are going to then most likely go to the cell membrane. So that's basically the cis face ends up being the input. So this is where proteins come in, and the trans face is the output. That's going to be the region where stuff is leaving the Golgi. There's your diagram and your electron microscope image of it, and we are going to stop part 1 of the video here. That way you guys can take a break and not be sitting in front of the, I guess I was going to say computer, but I guess your phone for so long.